So today we will be discussing with Dr. Vidya Niranjan her recent lab protocol article in PLOS ONE, Protocol for the Development of Coarse-Grained Structures for Macromolecular Simulation using GROMACS, and the associated protocol in protocols.io. Dr. Vidya Niranjan is head of Department of Biotechnology at the RV College of Engineering in Bangalore, India. Although she has dedicated the last decade to academia, she has also been in research and development sector in industry. Vidya has worked extensively on genome analysis, drug discovery, tools, and database development. With an extensive research experience of over 20 years, she has published over 100 research articles. She has also raised over $80 million of research funding from various government agencies and pharmaceutical companies. Vidya, thank you so much for being with us.
Thank you. Thank you for giving me this opportunity.
So to kick off this conversation, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yeah, so this is regarding the major challenge what the drug discovery company is going on, especially predicting the membrane and lipid proteins for drug screening. So now the existing all-atom model, which is computationally expensive and very popular, is not aligning with the necessary outcome, as most of the drug screening field is more on pharmacokinetics and thermodynamics, where based on that requirement, we decided that we will go with a coarse-grained model, which was published a few decades before, but I think we are using it right now to understand the drug screening process much better. So all atom model structures of biological macromolecules like proteins has to be converted into beads and run coarse-grained simulation using GROMACS is the novelty what we have started with. Though it is quite popular for some time, but we are using it for drug screening. And this coarse-grained simulations simplify the complex biomolecules and reduce computational cost, optimizing the study of larger system for a longer period of time. So this is why we are going for this simulation method. And also it plays a very vital role in drug screening for several reasons. So I want to just list it down. First is, already I have told, it's on computational optimization, which allows us to model larger and more complex molecular system, and also simplify the representation of atoms into interaction sites. So we all know that interaction sites are very important in drug discovery. So more than we consider as atoms, which is complex, now we are going to consider as an interaction sites. And we can also explore extensive conformational space and capture long timescale events that are often critical for understanding drug target and interactions. So this is something which I want to tell you, and as I already told, whereas binding kinetics and thermodynamics are utmost important, the coarse-grained simulation offer a practical solution. They enable us to efficiently study the binding events and conformational changes that might be elusive in traditional atomistic simulations. This is particularly significant when dealing with large biomolecular complexes such as membrane proteins and lipid bilayers, whereas the sheer size of the system poses computational challenges. Moreover, coarse-grained simulation provide a valuable tool for exploring the dynamics and flexibility of biomolecules, offering insights into structural changes that occur during drug binding. Their compatibility with enhanced sampling techniques further enhances our ability to capture rare events and gain a comprehensive understanding of underlying molecular process.
I think that you have very clearly explained not only what the protocol is, but what was the motivation to develop this coarse-grained protocol for simulations, and why previous protocols didn't work for you. But I would like to understand, when looking at the protocol itself, what are the most critical steps of your protocol? Those that the user really have to be careful.
Yeah, so actually, in this process of that, critical steps I would mention is, so I already told in the previous question that we are going to consider atoms into interaction sites, or what we call as a bead mapping. So this is very important step for to convert these atoms into beads, which is a most critical step. And this we have used using protein secondary structures. And that is something which is a very critical step here. And another one is the ligand parameterization. So whenever for drug discovery, the ligand has to be right now we have done with lot of trial and error methods. So these are the two critical steps that everyone has to understand before starting this protocol. And actually the motivations to optimize this protocol, because of in the field of this macromolecular simulation, the establishment of common and basic protocol for the development of coarse-grained structure is challenged for several key reasons. It ensures standardization across research endeavors, promoting consistency and the comparability of results. This standardization is essential for building a reliable foundation in the field. A common protocol also enhances data exchanges and format across different applications, tools, and compatibility among various simulation software and platforms. Researchers using different tools can adopt the same protocol, facilitating the exchange of simulation data and promoting collaboration across the scientific community. Moreover, the very important thing is the shared protocol reduces the redundancy in method development instead of each researcher independently creating their own coarse-grained methods, because this is going to be very tough and time-consuming. So this time and energy can be used in this can be used to refine and improve the common approach, so which will be more dedicated to the science community. So this is the reason that we have started this protocol for this method.
And since you mentioned time saving, that there is no reason for other scientists to start over, can you share with us if while developing this protocol, there was something that you tried but didn't work, and you decided not to include it in the final version of the protocol and the paper?
Yeah. You know that this is an in silico approach. So initially, before setting up this protocol, we have lobbied over many open source codes in Linux environment, which is similar, either in terms of, let us say, starting from coarse-grained model, Martin force fields, and then trying to adapt this in a Linux environment, and different versions where there is an incompatibility. So we have spent a huge time in first initiating this, though the GROMACS had this, but we also want to check with other GitHub, where especially because bead to mat, that is atom to bead mapping, there is no secondary structure automation is available in GROMACS. So we need to go through all these things, and also we need to find out this elastic parameters after setting up this model. So all these things were not easy for us, straightforward from the GROMACS. And ligand parameterization, all these three we need to lobby around various GitHubs, various tools, and so many things, Python packages. So I think our team has really put a lot of effort in making this work, and this workable version is already being done. And this is something which I want to save some time actually for the people in, though we have automized almost though we have automated majority of the things, but still we are having little more things to be added in future. That is the automate the upper cutoff of elastic network parameters, which we could not do that for this version. And finally, optimize the value using trial and error method. The team is really working hard on it. And also conversion and parameterization of the ligand structure could not be done, that still we have done manually. So these two things will be in our next protocol. I feel this will help the researchers to optimize their time and use it for science rather than really putting their time in fixing the softwares and tools.
I see. So that's where the protocol is heading. Those are the next optimizations and the next development that you want to do.
Yes, yes.
And now I would like to talk a little bit about your decision to have this protocol peer-reviewed as a standalone paper. So you share it in the open access platform of protocols.io, but you also send it to PLOS ONE for peer review. Why did you decide to have the protocol peer-reviewed?
Actually, peer review adds on additional layer of quality control to the lab protocols. Since I have expertise in publishing papers over last 6 years, very aggressively, over 100 papers, I think experts in the field assess the clarity, validity, and the feasibility of the protocols, helping to identify potential issues or ambiguities. This process enhances the overall reliability of the published methods when we are more focused on protocol development or tool development. Sometimes the scientists may not take some other dimensions from the input. So I think the different dimension input is also helpful for us to make this process much more mature. And we obtained actually a very important feedback to develop a detailed protocol for not only simulation of protein alone, but protein ligand simulation system, which has more application, obviously. And that is the reason we developed our protocol too. Actually, we had 2 protocols, which is published in PLOS ONE. So this gave us some more dimension and more innovation to work on, since we are already working on it. And the protocol have a wider scientific audience and application. It is most important for drug screening, especially if the target is transmembrane protein, usually a major setback in ALATM model. So we were also prompted and advised to show PCA-based thermodynamics properties by the reviewer in the expected results section, so that the readers know wide range of applications. So these are some of the things where the peer review has really helped us to come to think in a different dimension and work better.
I see. Thank you so much, Vidya. And I mean, you have an extensive and very successful career in science. And this paper was just published a few months back. So at this stage is just speculation. But can you tell us, what do you think the publishing of this methodological paper would have? What would be the impact of this paper on your career?
Yeah, actually, I have to really thank Protocol and PLOS ONE, where the thought process of the lab, like we started working on, initially it was just to optimize and just use the coarse-grained model for our regular drug screening data. But this optimization and making the complex problem to simple by using more of science domain knowledge and the technology, which is together, imbibed together, gave us actually it has given a big leap where we went into quantum computing. And fortunately, the Amazon and MIT, we had one of the project that has been sanctioned to give some space in quantum computing in Amazon bracket, where we just extended this in quantum computing and we found excellent results. So actually, which was running for at least a week simulation of these protein ligands. Now we are running it in hour or maximum couple of hours, which is optimized thing. Actually, a new dimension of the research using quantum computing is a real breakthrough. I hope it is going to have a major impact in drug discovery company or the societal impact, or something where this is going to have more impact. So we never, when we started this, we never had this, but publishing and getting reviews and from PLOS ONE and having that mind has helped us to get into drug discovery in quantum computing. And Amazon in India, we're very happy with us, because in the whole of this MAITI project, we were the one who started with quantum computing, and both the papers, one we have already published in protocol, and another two papers we have revisions, and we will be publishing it soon. So I think this is how we have taken advantage of our these particular protocol and PLOS ONE to to dive it into the next level, actually.
Thank you so much, Vidya. Your responses have been very, very thorough, very comprehensive, very informative. And I think that we cover many angles and many topics. But if there is anything that you would like to add to the conversation that I haven't asked, please go ahead just before we finish.
Yeah, I'm really thank you, and especially for scientists like us who are in some private institutes and who don't have much familiarity, but have a lot of aggression and interest in passion in science. We were not able to make this what you say is like this pedestal that you have given is making me feel better and more confident to do better, because such a nice opportunity to showcase my views, my research, and about the team and me is really helping and giving a lot of confidence for a women scientist who is in a private college. It's really good, and thank you so much for that.
Yeah, thank you, Vidya. Thank you very much.